it working. Thank you very much for coming. I know it's not easy at 5 o'clock. Welcome to this session. My name is Simon Schurz. I'm going to be the one who is chairing the session, which is dedicated to nanomedicine solutions for ophthalmology. We have 3 great speakers with us today. So I think that perhaps it's better that we just make the presentation and then dedicate like 10 minutes for questions, more than put all the questions at the end. Perhaps it will be easy for everybody. So let me introduce you the first speaker, Dr. Tina Wong, which is a senior consultant from the glaucoma service in Singapore National Eye Center in Singapore. Please, Tina, welcome. Thanks very much.
Oops, good. Good afternoon, everybody. Thank you very much for staying on to the bitter end. Actually, I only arrived here this morning, and it's 11 o'clock at night in Singapore, so I'm still here, so stay with me just for 20 minutes. I'm going to present a particular product that we've developed, which is using nanoparticles. Myself and my colleague, who was supposed to be here, he is on holiday, so he sent me instead. I'm the clinician, end of the story, so I'd like to take you through the journey of how we developed it. I'm a consultant ophthalmologist at the Singapore National Eye Center. My specialty is glaucoma, and here's my disclosure. First of all, let me introduce you to the eye disease that we are looking to treat with our nanomedicine. Glaucoma, what is it? It's a chronic and progressive disease of the optic nerve. And vision is lost through damage to the nerve due to elevated intraocular pressure. How is eye pressure produced? Well, there's a part of the eye where the fluid is produced in your eye called the ciliary body. It leaves through where you see the red arrow, through your pupil, and exits through the drainage angle. The cause of glaucoma is all about flow. What goes into the eye must come out, and if there's an inequality, you're going to have a buildup of eye pressure. And that's when you have damage to the optic nerve. Once the nerve is damaged, just like any other nerve in your body, you will lose the function of it, and as a result, in the eye, you go blind. What does it look like? Well, glaucoma starts from the outside in, so unlike age-related macular degeneration, where you lose the central vision, your central vision in glaucoma is preserved to the very end, and that is the problem with glaucomas, is that people do not know they have it until they have very advanced disease. In the world, by 2020, the WHO has estimated that about 80 million people will have glaucoma. And the current treatment is to lower the intraocular pressure. It's the only known modifiable risk factor. First line of treatment in glaucoma is eye drops for life. And the problem with eye drops, you may not just be using 1 bottle, you could be using 3 or 4 bottles, are the 3 points, which is bioavailability, the side effects, and patient adherence or compliance to medication. So first of all, let's talk about bioavailability. The problem with an eye drop, you put it in your eye, only about 5% is absorbed into the ocular tissues, and that is the therapeutic dose. 95% of it goes elsewhere. You know, you feel it at the back of your throat, it goes down your drainage duct, or it just runs on the side of your cheek. And there are also systemic absorption as well. Side effects. The other problem is that long-term use of eye drops can cause a lot of allergies, redness, itchiness, symptoms of allergies and long-term chronic use, because the eye drops, you know, with a bottle, you have to have it open for 30 days and you discard it. The reason why you can keep it for 30 days is because there are preservatives in the eye drop formulation, and long-term use of preservatives in the eyes chronically can cause toxicity. And this is a patient who was just on one particular eye drop for just 1 month, and he developed a severe allergic reaction. The other thing is that patients have to be able to put their eye drops in properly. Very often, people with glaucoma are elderly with arthritic hands, and they just cannot get the eye drops in, and so as a result, their disease can progress. One of the problems is compliance. We know that medical compliance is an important factor in controlling disease, not only in the eye, but any other systemic chronic disease such as diabetes, hypertension, you name it. There have been studies in the US that have shown that at least 10% of glaucomatous visual loss is due to poor patient compliance. And the most frequent problems that are faced by our patients is that they forget to take their drops. You know, even we, as healthy people, if you're given a 1-week prescription of, antibiotics, when you start feeling well, you start forgetting to take them. You know, I myself, I'm a doctor, I do that. So we're all, guilty of that to be compliant. And some people just completely stop taking it because they don't understand the disease or they don't think it's working. We did a study to see whether patients would actually prefer another way of taking the medicines apart from using eye drops, and this was like a little market survey, but while we were doing our research in the laboratory. We interviewed over 150 patients, and we asked them would they trade their eye drops for a more convenient way of delivering the medicines by a subconjunctival injection. And about 3/4 of them said that they would. The reasons? They were on more bottled medications, they were medicating more frequently, and they also admitted that they were non-adherent to their treatment regime. So we want to have a better extended release, more targeted release of a drug. And so what is on our wish list? Well, it has to provide equivocal therapeutic efficacy as eye drop formulation. It's got to be safe. It has to be optically clear, and that's why we went to a nano size, because if you're going to put anything into the eye, it's got to be small enough so it doesn't cause any visual problems at the visual axis. You want to have it as long as possible, and so for us, we wanted something that was at least 4 months in duration from a single application. It's got to be easy for the ophthalmologist or the doctor to deliver for the patients, and it's got to be comfortable and acceptable for the patients as well. How do we deliver drugs into the eyes apart from topical? There are various things. Some of them involve needles, which are scary. Some of them are like, underneath the outer layer of the conjunctiva. Some are implants, and some are reversible. Some are biodegradable. So there are various ways people have tried and tested delivering targeted delivery systems in the eye. So what we went for, primarily because my collaborator is actually an expert in liposomal technology, we looked at loading a particular anti-glaucoma drug using liposomes. And you know, the beauty of liposomes is that you have the hydrophobic outer layer, lipid bilayer, where you can load drugs, and also you have the aqueous core, the hydrophilic layer, where you can put your hydrophilic drugs, and so you can have some dual loading effect. Some of you probably already know this. The first nano drug which was ever approved by the FDA was back about 20 years ago now. It took 15 years to develop. And this was a liposome product which had like a coffee bean shape because the drug was placed in the center of the liposome, and it was able to be used as a cancer therapy. If you look at other liposomal drugs since then, have there been any that are extended release? Apart from Doxil, which is injected IV, so therefore there is an improved circulation time. But Deposit is the only other drug which is liposome-based, which has been shown to have an extended release of up to 5 days. If you look at this chart, there are presently about 12 approved liposome-based drugs available. About 25 of them are still in clinical trials, but there are 0 products in ophthalmology. So I think in a nutshell right now about sustained release and nanocarriers, the focus has always been on using nanosize to enhance cellular penetration or to enable prolonged circulation time in the IV administration. To date, there's no nanocarrier product that can sustain delivery of a drug for more than 5 days. The issue is really about loading the amount of drug you need to have sustained release, and then obviously to control once you load that drug, because you don't want that initial burst. So how do you retain the shape of your liposome with packing it full of drug? So that has been one of the major barriers for providing extended release beyond certain number of days. So as a result of that, there's been plenty of preclinical work, but not much success in the clinic. And this is just taken from a publication recently showing what the nanomedicine technology development pipeline, the timelines are to years of commercialization. And what we've been able to achieve under 5 years is from the basic nano research, proof of concept, preclinical studies, to the first in man. We reached this in 5 years, which is actually quite an incredible feat by standards of nanotech development. So this is our product. It's the drug liposomal latanoprost. Latanoprost is a prostaglandin analog. It is actually one of the most successful drugs which was developed by Pfizer about 20, 25, 30 years ago now, and it is very effective at lowering the intraocular pressure. So there are some comparative features of the latanoprost eye drop. On the left, it's marketed as the name Xalatan, and our liposomal latanoprost or lipolat formulation on the right. You can see a single drop in volume. We can load over 12 times amount of latanoprost drug in our liposomal technology. The lipids we use are neutral. There are no preservatives, and the pH is comparable to the eye drop formulation. Therefore, it's compatible with injection into the eye. We then, after we did all the bench testing, we did extensive, prolonged preclinical studies on 2 animal models. We used the New Zealand white rabbit, where we saw a pressure lowering of up to 4 months from a single injection. Then we did repeat injections to show that it was safe. You can see on the left is that there's the procedure being done in the animal, just held with forceps with a 30-gauge needle. You're injecting on the conjunctiva. The second image here is just showing you the bleb where the lipolat has been injected. After about an hour, this blister disappears and the drug is absorbed. Here you can see that this is the same drug, but it's tagged with marina blue dye so that you can see where the liposomes are when you shine UV light. And this is actually just to show the presence of the drug. And within 2-3 hours, the bleb has disappeared and the eye is nice and white. The other thing is that we evaluated our drug compared to monkeys, macaques, and you can see that we compared it with the eye drop formulation, and with 1 injection, the pressure lowered. And in the eye drop group, the animals received the eye drop on a daily basis. So you see the pressure lowering effect. The pressure dropped from 20 down to about low teens, and it was retained for up to about 120 days with the liposome formulation. Then the pressure started going back up, so we re-injected, and we could reproduce the same effect. So given that we were able to replicate this, and we did extensive safety studies in animals, we then proceeded to conduct a clinical study. This was a first-in-man study, a phase 1b study on 6 patients in Singapore. We have got it approved through our local regulatory body, and this was an open-label study on 6 patients who had glaucoma. This was a safety study initially, primarily with secondary outcome of efficacy. Patients were followed for 3 months. They had to be already on an eye drop, 1 eye drop, and we took them off their eye drop for 1 month to wash out the effects of the eye drops before we gave them the injection of lipolat. So this is what we did. We gave them a 100 microliter injection, just like you saw with the rabbits. It's the same volume, and then we measured the intraocular pressures throughout the 3-month period. So the next one should be a movie just to show you the injection that we performed on our patients. You see here is the needle going in. This is with the eyelid lifted up and the patient looking down. And there is no pain or discomfort. We gave them some topical anesthetic eye drops to start with so that they would not feel any discomfort. And this is 100 microliters injected in the patient. So the results, patients were well, they tolerated the injection. There was no problem. 2 patients actually experienced some dry eye symptoms because we took away their glaucoma eye drops, and so they started experiencing their dry eye, and so we prescribed lubricants, and that settled their symptoms. Apart from that, there were no other safety issues or any evidence of inflammation or infection at the injected site. If we look at the intraocular pressure lowering, it was very comparable to the animal data, which was very much to our surprise. We actually found that there was a clinically significant intraocular pressure lowering. Now, I just want to focus that the clinically significant IOP reduction has to be at least 20% reduction from baseline intraocular pressure. We were able to achieve more than 30%. Here you can see very clearly the mean percentage IOP change from baseline. Immediately after injection, you had about 50% reduction in intraocular pressure. And then throughout the 3 months, we were able to achieve at least 30% reduction, and it was pretty steady, and we were very, very pleased with that outcome. So I'd like to summarize that we were able to achieve, develop a new extended release formulation of an anti-glaucoma therapy using nanomedicine or nanoliposomes. We can achieve a clinically significant therapeutic pressure lowering effect. The Lipolat was very well tolerated by all patients, and there were no real substantial safety issues, at least for the first 6 patients. There were no serious events or sequelae from the injection, and we feel that this is a promising and feasible way of delivering drugs to treat patients. So I'd like to just conclude that we've been hearing about how it is difficult to get to approval for certain drugs, but I think it's a matter of matchmaking the right nanocarrier with your drug, whatever you choose. We were very lucky; it was an off-patent drug, and we tested a lot of liposomes, but we eventually ended up with the right one with the drug, and it matched very nicely. And from that, everything else went very quickly. We were actually funded very well by the Singapore government, but we were able to conceptualize, bench test, formulate, develop all the preclinical, clinical testing, and get approval within 5 years. So I would like to say that also it's the first nanomedicine for ophthalmology. We were able to achieve it. And it is also a landmark achievement in terms of extended drug release. It can be done for nanomedicines beyond 5 days. We've been here, I'm standing here, and I'm just able to share this with you to prove that it can be done. And as a result of that, this technology has been licensed to a spin-off company, for which I'm also the co-founder and I'm the CEO of. And we're now taking it to the next level of multi-center trials, and we're about to submit an IND at the end of this year. So I'd like to thank everybody and the organizers for this opportunity to speak to you. And also I'd like to just acknowledge all the teams from the Research Institute, from the university, and also from the government for all their funding. Thank you.
Thanks very much. Questions?
Wonderful work. Did you look to see whether or not you had a short-term disruption of the tear film after the injection from some of the drug leaking back out? Have you looked at any of that yet?
No, we didn't really look at that. We actually used this as smallest caliber gauge needle as possible. We had it when, I don't know if you saw, but when I finished the injection, I held the needle in there for a little bit before I withdrew to limit that. Yeah.
Thank you. Have you any experience with patients underwent surgery? So would this medication also be efficient in patients having some surgery already on glaucoma?
Yes, as long as they're on medication. And sometimes surgery doesn't work that well and they still need medications. So they would still be able to, I would see that anybody, yeah.
So scarring and so on,
Yeah.
it's now hindrance.
Yeah.
Okay, thank you.
Have you seen any systemic effect of the drug?
No. We've done talks, extensive talks on the animals. We haven't seen any effects. Thanks. Thank you.
Penetration of the drug into the different compartments of the eye, and specifically the posterior segment, the vitreous cavity.
Yeah, thanks for that question. I'm glad you brought it up. Actually, with one of the photos of the rabbits, which was with the UV light, we actually were curious to know where it all went after an hour. And it actually penetrates into the posterior segment, is resident in the vitreous, and it stays there for at least 80 days. So we know that you have an extended release platform in a reservoir, the vitreous, which is another extended release platform. So that might explain why we're having such a profound and prolonged effect in the eye, because the eyeball itself is actually a privileged site. Whatever you inject there doesn't really go anywhere else systemically, unlike if you're trying to target the liver or the pancreas. It doesn't have to go IV. Yeah. No, we don't. No, we don't need to. Yeah.
Here. What type of liposomes?
Yeah, we used egg PC.
APC?
Egg PC.
Only APC?
Only egg PC. But the new formulation, we're using a pure form, so we're just using pure PC. You know that egg PC is the major lipid in egg PC is pure PC.
And which was the concentration that you injected? The lipid and the
1 mg per ml
drug?
is 1 mg per ml. Yeah, of
And,
latanoprost, and it's 100 microliters.
Okay, thank you.
Okay.
When you showed your data for your human trials, you had a very sharp decline in the intraocular pressure after the injection. Is this very sharp decline in the beginning due to some burst release from the liposomes, or is this because of the injection itself?
Oh, okay. That's a very good question. I didn't show any in vitro data because it was just too much. But the in vitro data shows no burst. It's actually very linear. The reason why you have a very rapid reduction immediately after injection is because it causes some vasodilatation. Because the drug is a prostaglandin, and prostaglandins are in the pathway for inflammation like leukotrienes. So you do have, upon injection, you have a bit of vasodilatation that allows the drug to penetrate very quickly. And so that's a result of its immediate action. So then after it drops down, it comes back up. When the vasodilatation stops physiologically, you have a stabilizing effect. Thank you.
Questions? We go to the next speaker.